How's it going, everybody? Herman here again. Today we're going to be talking about inheritance. So let's jump right into it. So we have epigenetics, which is actually something that we've already covered several times in this course already. We're going to talk about sex chromosomes, linkage on the chromosomes, non-disjunction, certain types of, I guess we could call them as failures that happen on the chromosomes, sex determination, how that's determined, and X and Y linked traits. So, like I said, we've already discussed epigenetics before. We know that it's the study of inherited changes in a gene, with an expressed phenotype that happens without changing the underlying DNA sequence. So, epigenetics plays a crucial role in regular gene expression. Like I said, we discussed this already. We've talked this over at least 2 or 3 times now. So there are mechanisms for certain things that happen during epigenetics. One of those is DNA methylation. So the addition of a methyl group to DNA, and it typically results in gene splicing. Histone modification, another form of epigenetic transformation or genetic change that doesn't change the DNA sequence. And this is the addition of various chemical groups to histone proteins, which can either relax or compact chromatin. So chromatin is the reorganization, or chromatin remodeling is reorganization of chromatin structure, allowing or blocking access to transcription factors. Epigenetics regulates gene expression through gene slicing, gene activation, cellular differentiation, and gene slicing epigenetic mechanisms can silence gene expression by preventing transcription. And gene activation epigenetic mechanisms can also activate gene expression by allowing transcription. So just the vice versa effect. A crucial role in cellular differentiation allows cells to adopt specific functions. Some factors that influence epigenetic regulation of gene expression. Obviously, we know environmental factors plays a major role in regulating gene expression. So diet, stress, household upbringing, all these things can influence epigenetic marks. Some developmental factors. We've discussed cellular differentiation. All these influence epigenetic marks. Mutations can also influence epigenetic marks. Some techniques that are used to study epigenetic regulation. So we mentioned DNA methylation. There are techniques such as bisulfite sequencing, and this is used to analyze DNA methylation patterns. This was one that I was familiarized with CHIP when I took immunology. Techniques such as CHIP sequencing can be used to analyze histone modification and chromatin remodeling patterns. And CHIP stands for chromatin immunoprecipitation. Gene expression analysis techniques such as RNA sequencing can be used to analyze gene expression patterns. Applications of epigenetic regulation of gene expression. We have cancer research, neurological disorders, developmental biology. One second. Sorry about that. Epigenetic regulation of gene expression can provide insights into cancer development and treatment. So sex chromosomes determine the sex of an individual, and there are two types of sex chromosomes. We have X chromosome and the Y chromosome. So this also determines sex in a lot of types of species, in humans as well. So females have a XX chromosome, males have an XY. Most species, sex is determined by the presence or absence of those specific sex chromosomes. There's also some species that their sex is determined by environmental factors. We'll talk more about that later. X-linked traits are characteristics that are determined by genes locations on the X chromosome. These are inherited in X-linked pattern, where males are more likely to express a trait. X-linked recessive traits are expressed in males who have only one X chromosome. X-linked dominant traits are expressed in both males and females. Y-linked traits are determined by genes located on the Y chromosome. Y-linked traits are inherited in a Y-linked pattern, where only males can inherit this trait. Y-linked traits are quite rare, as the Y chromosome is small and contains very few genes. Some abnormalities occur when there's an abnormal number of or structure in the sex chromosomes. There's abnormalities in structures in the counts. We have Klinefelter, Turner syndrome, XYY syndrome, and that's a condition where males have an extra Y chromosome. We'll talk some more about these syndromes here in a little bit. X-linked traits are characteristics that are determined by genes located on the X chromosome. So just a little bit to review. X-linked traits are inherited in an X-linked pattern, where males are more likely to express a trait. X-linked recessive traits are expressed in males who have only one X chromosome. X-linked dominant traits are expressed in both males and females. Some examples would be hemophilia, a genetic disorder that affects blood clotting caused by a mutation in the F8 or F9 gene on the X chromosome. Red-green color blindness, a condition where individuals have difficulty distinguishing between red and green colors, and this is caused by a mutation in the OPN1LW or OPN1MW gene on the X chromosome. Duchenne muscular dystrophy, a genetic disorder that affects muscle strength and function, is caused by a mutation in the DMD gene on the X chromosome. Here's some examples of Y-linked traits. Male pattern baldness, a condition where males experience hair loss caused by a mutation in the AR gene on the Y chromosome. Acne, skin condition characterized by pimples and inflammation, influenced by genes on the Y chromosome. Height is influenced by multiple genes, including those on the Y chromosome. And a little bit of comparison. X-linked traits are inherited in the X-linked pattern, while Y-linked traits are inherited in a Y-linked pattern. X-linked traits can be expressed in both males and females, while Y-linked traits are only expressed in males. X-linked traits are more common than Y-linked traits, as the X chromosome contains more genes than the Y chromosome. So linkage refers to the physical connection between genes on the same chromosome. So we talked about how distance tends to influence certain types of genes. I think that's one of the key concepts that we have to keep in mind as we continue to talk about chromosomes and genes. So these genes are physically close together on the same chromosome, and that is what we refer to as linked genes. They can be separated through recombination, and this happens during meiosis. A linkage group are genes that are grouped together. Then there's types of linkage coupling, or linkage groups. So when two linked genes are inherited together, more often than not, they're expected to. It's chance. It's all by chance. Remember that when these chromosomes are aligning on, I keep coming back to this, but on the metaphase plate, like there's literally millions of combinations. And I don't know if I mentioned this last time, but a perfect example would be like my nieces. So one of them was born with a darker skin tone, kind of like me, and then my other niece, she has her mother's skin tone, which is a very light, almost a pale skin. It's not that anything funny happened there, right? It's just simply the way the chromosomes line up at the metaphase plate. There's just so many combinations that these genes can take. So yeah, a lot of these genes happen just by chance. It's all happenstance. I do believe they're working on certain types of genetics as far as reproducing, where they can create the traits or have the phenotype expressed, a specific phenotype expressed. Repulsion occurs when two linked genes are inherited together. And this is all by chance as well. So distance between genes on a chromosomes highly affects the likelihood of recombination. The frequency of recombination between linked genes can vary. Chromosome structure, such as the presence of chromosomal inversions, can affect linkage as well. Linkage maps are used to visualize the arrangement of linked genes on a chromosome. Recombination frequencies are used to estimate the distance between linked genes. And LOD scores are used to measure the likelihood of linkage between genes. Understanding the linkage is important for genetic counseling, as it can help predict the likelihood of inheriting certain traits. This is especially true for inheriting certain diseases, right? Certain diseases that are passed by either the mother or the father's genes. Linkage mapping is also used to identify the location of genes on a chromosome, and this is like one of the gene cloning is one of the things that I mentioned, how understanding linkage can facilitate gene cloning, as it helps identify the location of a gene of interest. So this is what I'm talking about. People look at gene cloning in order to express the phenotype that they want. I know for certain in my family we have pale skin and we have colored eyes. So I don't know, perhaps that's something that one of my descendants or one of my somebody in my family could possibly look into one day. Me personally, I have a high or above average height, and of course above average looks, as you can see. So let's go over some of these terms again. So the distance between genes on a chromosome affects the likelihood of recombination. The frequency of recombination between linked genes can vary. Chromosomal structure, chromosomal inversions can affect linkage. The linkage maps are used to visualize arrangements of linked genes on a chromosome. And so the frequency of recombination um, is used to estimate the distance between linked genes. Lot scores are used to measure the likelihood of linkage between genes. Pedigree analysis is used to study inheritance of linked genes in families. Like I said, people can go back and look at the certain phenotypes that they want to see, or maybe they're just genuinely curious. Linkage disequilibrium is used to study the association between linked genes and populations. QTL mapping, quantitative trait locus, is used to identify the location of genes that contribute to complex traits. We have they've developed software. A lot of this, most of these things, as far as genetics goes, it's all digital. We have ELISA, we have Western blot. So I found some software linkage analysis that they use. This one's actually called linkage. It's obviously it's to analyze linkage properties. CRI map is also used for linkage mapping. MapMaker linkage mapping. There are some limitations to this. As I said, there's always a lot of these things are by chance, but recombination frequency can vary different between regions on chromosome, or it can vary greatly between different regions of chromosomal regions. Genetic heterogeneity. It can make it difficult to identify linked genes, and the sample size. It can also affect the power of linkage analysis. So now we're going to talk about non-disjunction. Non-disjunction is the failure of chromosomes to separate properly during cell division. It results in abnormalities in the number of chromosomes in the daughter cells, and this could lead to a lot of issues. So non-disjunction that occurs during meiosis can result in a number of chromosomes or an abnormal number of chromosomes in the gametes, when that being the sperm or the egg cells. So you already have issues from the get-go. If this occurs during mitosis, resulting in an abnormal number of chromosomes in the somatic cells. So, I mean, errors happen on the chromosomes. Like I said, think about this is happening, as far as mitosis go, I mean, this is happening all the time in our bodies, right? Just the probability of something going wrong is always extremely high. Good thing for our cells is that they can recognize when one of these cells, comes out a little wonky, and they quickly proceed to destroy it. Your white blood cells are predators; they are deadly, and that's why when they get sick, things can get pretty bad. But this is fairly common; it happens all the time. But our cells are very powerful at detecting these kinds of errors. I think they even kill innocent cells as well, if I'm not mistaken. There's a lot of killing that goes on within our bodies. Some chromosome abnormalities, such as translocations or deletions, can increase the risk of non-disjunction. And as I stated before, I mean, this could even happen, when your DNA is separating, or even when an RNA molecule is being coded. Like, there could be a deletion somewhere or an insertion, and then all of a sudden you have a sick RNA strand. Like the possibilities they keep compounding. They keep compounding. But like I said, our bodies are quite adapted to these things, and particularly, what is it, beta cells, when they give rise, the way that they get detected it's incredible mechanisms at work. Environmental factors always come back. Exposure to toxins, radiation, all this can increase the risk of non-disjunction. Um, Genetic factors such as mutations in genes involved in chromosome separation can also increase the risk of non-disjunction. I think even certain types of mutations can help as well. You know, it's not always a negative thing. They say this all the time. If a woman is of an older age, I believe after 40, a woman starts running risk of developing age-related chromosomal factors, and this all leads to non-disjunction. It's really what they mean is that whenever these chromosomes are aligning, I don't know what happens to the woman. I guess these guys are maybe a bit slower, but things tend to give problems tend to arise when a woman's age comes into factor. Um, so this can result in aneuploidy, and this is when an individual has an abnormal number of chromosomes. That's all it means. And this leads to birth defects, infertility, miscarriages, even cancer. Um, some of the birth defects, I mean, the most common one would probably be Down syndrome, where there's an extra chromosome and this ends up completely, um, usually rendering the offspring um, at risk, of Down syndrome. You also have Turner syndrome, infertility. Non-disjunction can also increase the risk of infertility. Miscarriages are always at play here. These are some of the ones that tend um, be related um, to women's age, and so chromosome 21 uh, is Down syndrome results when chromosome 21 results with an extra copy of that particular chromosome. Turner syndrome is caused by non-disjunction of the X chromosome, resulting in a missing or partially deleted X chromosome. There's also Klinefelter, which is caused by non-disjunction of the X chromosome, resulting in an X chromosome in males. We have trisomy 13, 18, and this is in regards to the specific chromosome, which is chromosome 13 for trisomy 13 and 18 for trisomy 18. Karyotyping is a test to diagnose chromosomal abnormalities. So there are ways that they test this now. FISH is something that I've seen before in, I believe it was in immunology. It's a test to diagnose chromosomal abnormalities as well. It's basically fluorescence in site hybridization. Microarray analysis is a test used to diagnose chromosomal abnormalities, including non-disjunction, prenatal testing, villus sampling, and this is all to diagnose chromosomal abnormalities. PGD, pre-implantation genetic diagnosis, um, it checks for chromosomal abnormalities in embryos created through in vitro fertilization. So that is actually really cool stuff. Remember how I was talking about how certain parents want certain traits for their children? This usually happens in vitro fertilization. Um, so, I mean, there are ways to, I guess, sort of prevent these sort of abnormalities from being dealt with or being a part of someone's life. Um, so we have genetic counseling. When people, you tell them the risk. Does the benefit outweigh the risk? If you're talking to parents that are already, of age, let's say they're 50 years old and they're considering having a child, these things could be talked about. Prenatal testing, making sure that you're making a properly informed reproductive decision. Um, PGD, again, they can test whenever in vitro is involved. Uh, Healthy lifestyle. I mean, healthy lifestyle is going to be prevalent in any facet of biology, um, and also not exposing yourself to toxins. It's funny. I was talking to this woman from Venezuela, right? And she told me the city that she's from, the lake is actually contaminated. And so it's just something funny, you're not going to go jump in that lake and swim around and, not expect to develop some sort of environmental toxin within your body. And it could eventually, lead to reproductive defects. So just taking care of yourself, making wise decisions. Don't jump into the lake in Maracaibo because you'll get sick. um, Prenatal care. I know they have these sort of vitamins that women take. Um, even emotional support. Emotional support can play a major role in connecting individuals who have experienced non-disjunction with someone who hasn't experienced yet. The medical treatment genetics right now is at a far advanced level. Unfortunately, it is very expensive, but there are many alternative methods in order to achieve a person's goal as far as reproduction goes and non-disjunction. Um, for those that are interested, research is always a great way to to enter an area that is possibly not underdeveloped, but could use some extra hands. But researchers are studying, the causes of non-disjunction, and this includes factors that we've talked about being genetic, environmental, emotional, psychological, all these factors that play into non-disjunction and its effects. Um, they're working on just seeing the results of what non-disjunction does, and that being in the lab or in a household. Um, so they're developing methods for preventing and even treating non-disjunction. This includes genetic testing and gene editing. I believe gene therapy has the key to non-disjunction and many other diseases. Like I said, advances in genetic testing and gene editing may enable personalized medicine approaches to specific uh, individuals in order to prevent and treat non-disjunction. Um, so in conclusion, non-disjunction is a complex and multifaceted phenomenon, and it can have significant consequences for fertility and health. Understanding the consequences of non-disjunction is essential for developing effective prevention and treatment strategies. And there's still ongoing research, developments that um, need to better to understand non-disjunction and improve outcomes for individuals affected by this condition. So now we get to the final topic, sex determination. And this is the process in which individual sex is determined. Individual organisms, specimen. Um, there are different types of sex determination. So we have a genetic sex determination, which is the most common type. It is the presence or absence of a specific sex chromosomes, and this determines an individual sex. We know, of course, in women and or humans, it's XX and XY. Um, Environmental sex determination. So this occurs in some species where environmental factors such as temperature or social factors determine an individual's sex. Um, I believe a specific one would be seahorse, if I'm not mistaken. It's very interesting stuff going on at work there. Hormonal sex determination occurs in some species, and this is where hormones such as testosterone or estrogen determine an individual's sex. Um, X and Y chromosomes. So in humans and other mammals, the presence or an absence of the X and Y chromosomes determines the individual's sex. Right, I just mentioned that. In humans, other mammals, individuals with two X chromosomes are female, while individuals with one X and one Y are male. The SRY gene located on the Y chromosome plays a crucial role in sex determination, triggering the development of testes in males. Imagine if something went wrong there. That would be quite traumatic. Some species, such as turtles and crocodiles, the temperature of the incubating eggs determines the sex of the hatchlings. Some species, such as clownfish and zebrafish, social factors such as dominance hierarchies, all determine the individual sex. Hormones such as testosterone and estrogen play a crucial role in sex determination, regulating development of reproductive organs and secondary sex characteristics. Believe it or not, that actually does play a role in humans as well. Um, I believe it leads to certain feminine characteristics in males. That's why you have some males that are not as rugged, on the outside as another male would be. Um, Again, millions of possible things could happen as an offspring or zygote is formulating, right? And that being in the womb or in the lab or in a in an enclosed capsule. Individuals with androgen insensitivity syndrome, the body is unable to respond to testosterone, resulting in the development of female secondary sex characteristics despite the presence of a Y chromosome. So exactly what I just mentioned, that's just something that could go wrong. A guy is born with feminine characteristics. And not always is this determined, how would I say, um, the lifestyle, right? Like this person could be very much interested in the opposite sex, the opposite gender. It's just some of these things that happen. We mentioned Turner syndrome. It's where females have or partially deleted X chromosome. They're missing it or it's partially deleted, and this results in infertility and other health problems. Birds, sex is determined by the presence or the absence of Z and W chromosomes, with males having ZZ chromosomes and females having ZW chromosomes. In fish, sex is determined by a variety of mechanisms. This includes genetic, environmental, hormonal factors. I think fish are probably, or what would you consider a fish? Is a starfish a fish? I guess it's in its name, right? Seahorse, a fish. Um, a whale is not a fish, right? It's a mammal, but that's kind of strange. But I mean, all these factors come into play. I'm telling you, check out seahorses. It's actually really cool how they reproduce. Insect sex is determined by a variety of mechanisms, including genetic, environmental, and hormonal factors as well. Um, the origin of sex chromosomes is thought to have occurred around 300 million years ago, with evolution of the X and Y chromosomes in mammals. Sex determination mechanisms have evolved independently from different species with a variety of genetic, environmental, and hormonal mechanisms being used. Sex chromosome abnormalities such as Klinefelter-Turner syndromes can have significant clinical implications, including fertility and other health problems. Intersex conditions where an individual has both male and female reproductive organs. That's one of the nightmares that could happen. Can be caused by a variety of genetic and hormonal factors. I think hormones play a major role in that. Sex assignment, the process of assigning the sex to an individual at birth, can be complex and it may involve a variety of medical and psychological factors, right? Like nowadays, nothing is linear anymore. I feel like it never really was, but well, I guess we've finally come to accept it. I mean, nothing is linear in life. Nothing is 100% certain. This great man made a great analogy. A lot of times we're so trustworthy of like medications that we're taking that medication was made, you know, um, under the right chemistry. Generic materials weren't used. The addition and removal of chemical groups was done down to a T. The organic chemistry was there. Um, but you never know. Something could have gone wrong, and it could have been just that one vial of pills that we get, right? But nothing is linear. I think this is a great topic to discuss and to talk about. But yeah, those are some of the clinical implications of uh, intersex and sex assignment conditions. But there is ongoing research. Discussion is heavily needed to address the complex issues surrounding sex assignments and intersex conditions. So that's all I got for you guys today. Hope you guys have a wonderful day if you're watching this. It was a pleasure, and I'll see you guys in the next video.